This tutorial will focus on gene regulation. That is, how do cells go about regulating the functioning or expression of genes? The expression of a gene refers to turning the coded information on the gene into a protein. That's what we mean by gene expression. The code is expressed as a protein. Most genes need to be either switched on or switched off to function, and in many ways many genes aren't switched on all the time. There's all sorts of reasons why a gene may not be switched on all the time, but to have genes switched on all the time would require energy, because that would mean that proteins would be being produced all the time, and cells don't want to waste energy, so they've evolved to conserve their energy. So genes will be switched on and off to conserve energy. But of course, there are many proteins that a cell doesn't want always manufactured all the time. Production of enzymes has to be stopped at certain points. At other times, enzymes have to be switched on. Structural proteins can't be made all the time. So you can't have genes switched on all the time, otherwise you'd have this uncontrolled production of proteins. And the production of proteins has to be controlled. If protein production has to be controlled, so does the functioning of the genes have to be controlled. And this is what we mean by gene regulation. And this is a very complex process in terms of regulating genes. This diagram on this page shows one of the early understandings of a prokaryotic gene regulation, and this is an example of the gene that controls the production of an enzyme called galactosidase, which is an enzyme that helps to break down the sugar galactose. It was a very early discovery in terms of gene regulation. I'll just show you this diagram just to give you a bit of a sense of the complexity of regulating a gene, and also to introduce you to a few terms, and then we'll look at a couple of other concepts in relation to this. The gene that we're interested in, a gene that will code for a particular protein, a coding gene for a particular protein, is referred to as a structural gene. We can see that shown here. So the structural gene, this segment of the DNA, codes for the enzyme that the cell is interested in this case beta-galactosidase. So this structural gene has three segments, and it leads to the production of three different enzymes that are all related to metabolizing galactosidase. Metabolizing galactose. So this is what we refer to as the structural gene. It's the one that's going to be responsible for the production of the proteins that are going to do something in the cell. But to make the structural genes work, that is to switch them on, involves a whole lot of other segments of the DNA as well, and these are referred to as the regulatory sequences, and you can see this here. And this effectively would be a number of the the introns that we talked about in another tutorial. Of the regulatory sequences, there are such things as a section of the DNA referred to as the promoter. The promoter is a part of the DNA that helps to regulate the production of another protein called a repressor protein. So you have a regulatory gene noted here. So we have a gene that is going to code for a regulatory protein, in this case a repressor protein. So this section of the DNA is referred to as the promoter for the regulatory gene, so it helps to regulate the regulatory gene. This is the regulatory gene here, which will code for a repressor protein. To repress something means to stop it, so this is a protein that is going to stop something happening. So the regulatory gene codes for a repressor protein. Then you have another section of the DNA which is the promoter for the structural gene. And you remember the structural genes are over here. And to make the structural genes work, you need an operator gene, and that's located here. So you can see, even in this what they call simple example, it sounds very, very complex. So to make the structural gene turn on, you need to have the promoter for the regulatory gene itself, and the promoter for the structural gene and the operator gene all working in harmony to switch the structural gene on, which will produce the enzyme of interest. Let's have a look at how this might work in practice. Again, complex diagram, don't get too fazed by it, but have a close look at it just to see how different things are happening. Again, you'll probably find many animations on YouTube which will help to show you how this works in practice. So let's have a look at how this might work. This is an example of a structural gene that's going to involve something to do with enzymes that relate to lactose. So the structural gene or genes are up here. They're the ones that are going to produce the protein of interest. But to get those structural genes switched on, other things have to happen. So here's our regulatory gene, further down the DNA. The regulatory gene will go through transcription and translation to produce a protein, in this case a repressor protein. We can see that here. Now, what the repressor protein does is when it's made, the repressor protein binds to the DNA around the operator region. So it blocks the operator. So while the repressor protein is blocking the operator, that will switch the structural genes off. So the structural genes cannot be transcribed while the operator is blocked by the repressor protein. So this is a way of switching the structural genes off. So the RNA polymerase shown here, which will be would normally be moving down the DNA to make messenger RNA from the structural genes, cannot move past the blocked operator here. So the gene is switched off. How might it be switched on? Well, in this case, the gene is ultimately switched on by the presence of the ligand, in this case lactose, in the environment of the cell. So the presence of allolactose, in this case, induces the repressor protein to unbind from the DNA. Let's look at this step by step. So up here, the gene is switched off because a repressor protein is blocking the operator, shown here. Let's have a look. So the regulatory gene, shown here, is producing a repressor protein, which would normally bind to the operator, here. But in this case, when allolactose is actually present in the environment, this molecule, lactose, binds to the repressor protein and inactivates it. In other words, it prevents the repressor protein from binding to the operator. So the repressor protein cannot bind to the operator. That means that RNA polymerase can continue its way down the DNA, and it can then transcribe the structural gene here into whatever the appropriate enzyme or protein it happens to code for. If the allolactose here was to disappear from the environment, imagine what would happen. So the lactose disappears, the repressor proteins that are made here would no longer be blocked, and they would again bind to the operator and block the operator, and that would again switch the structural gene off. If lactose was present in the environment again, again it would bind to the active repressor, inactivate the repressor. This would unblock the operator here, and it would switch the structural genes on. So what switches these genes on, structural genes on and off, is, in this case, the presence or absence of allolactose in this particular example. And I imagine that the protein that would be made from this would be something that acts on the lactose itself. Now this seems incredibly complex, but scientists working in this field would say this is the bare basics. This is the simplest version of gene regulation. This is in bacteria and relatively simple. In complex eukaryotic organisms like ourselves, gene regulation is often a far more complex affair. But if you had this basic idea that there are structural genes, regulatory genes, promoters and operators and all these work together to switch genes on or off, then you have a sense of what gene regulation is about. Linked to this topic is the idea of master genes. Sometimes, or not sometimes, they're referred to as homeotic genes, and you'll have taken notes on this. Homeotic genes. They're almost like a master gene, the code almost for a blueprint, if you like, in the embryonic development of animals. So rather than a whole lot of individual genes that produce code for a whole lot of individual proteins, these master genes, or homeotic genes, control the development of multiple parts of the body all at once. Since the discovery of these, a lot of things in evolution make a lot more sense. If you could imagine that there was a mutation or mistake or change in a master gene, then you could see a fairly significant change in a body part, a significant body part, fairly quickly. So rather than lots of many tiny little minor changes because of mutations, you could see a significant change happen very quickly. There's a wonderful photograph here, or maybe not wonderful when you look at it, but it's rather astounding, of a mutation that occurred in a Hox gene, which is a kind of homeotic gene. So Hox genes are a kind of homeotic gene. They're a master gene that controls segments in particular insects. And a mutation, which is a mistake in the working of a Hox gene, in this photograph has led to an extra leg growing on the head of this insect. So we actually have the head, so we have an eye, and an eye. This is the head of the insect, but most unusually you have a leg that's growing out of the head of the insect. In other words, due to a mutation in a master gene, you've got a whole body part that's growing in the wrong place. So this is not some kind of minor mistake. This is a significant mistake in the way that the Hox gene works. So homeotic genes, which are these master genes that control development of whole sections of organisms in the embryonic stage, are significant in terms of the regulation of the way bodies work and the way bodies are built.